Welcome back to AI and Stuff, where the future of science is starting to look less like one exhausted researcher with 97 browser tabs and more like a human scientist, plus an AI research team. This story is about Robin, a multi-agent AI system from FutureHouse. Nature published the paper on May 19, 2026, under the title A Multi-Agent System for Automating Scientific Discovery. And yes, the system includes agents named Crow, Falcon, and Finch. Science finally got AI assistants and immediately named them like a tiny research squad. Robin is exciting because it does not stop at reading papers. It connects more of the discovery loop. It searches literature, proposes experiments, waits for human scientists to run the real lab work, analyzes the data, and suggests the next experiment. That is the pro-AI story. AI is not just chatting. AI is becoming scientific infrastructure. Think of Robin like a lab partner that says, here is a testable idea, here is why it makes sense, here is what the data shows, and here is what we should try next. The disease they focused on was dry age-related macular degeneration, or dry AMD. Dry AMD is a major cause of vision loss, especially in older adults. Simple analogy? Your retina is like the camera sensor in your eye. Behind that sensor are support cells called retinal pigment epithelial cells, or RPE cells. One of their jobs is cleanup. They eat cellular waste through a process called phagocytosis. If that cleanup slows down, waste can build up and vision can suffer. So Robin asked a practical, useful question. Can we find drugs that help those cleanup cells work better? According to the preprint, Robin first reviewed 151 papers to propose disease mechanisms and assays. Then it reviewed about 400 more papers about RPE phagocytosis and dry AMD treatments. From that, it proposed 30 existing drug candidates for testing. That is where AI shines. Not because it is magical, because it can search a huge, messy knowledge space and hand humans a shorter, smarter list. While a person is still trying to remember where they saved the PDF, Robin is already building a testable candidate set. Researchers then tested top candidates in RPE cells using a phagocytosis assay. The test is easy to picture. Give the cells a drug, give them tiny fluorescent beads to eat, and measure how much eating happens. One early winner was Y27632, a ROCK inhibitor. ROCK inhibitors affect the cell's internal scaffolding, which can make it easier for cleanup cells to move and swallow debris. Then Robin did something that makes this more than a fancy search engine. It suggested a follow-up RNA-seq experiment to see what changed inside the cells. RNA-seq is like checking which instruction pages a cell is reading more loudly or more quietly. Finch, the data analysis agent, analyzed the results. The paper reports a threefold upregulation of ABCA1 in Y-27632 treated cells, with an adjusted p-value of 2.13 times 10 to the minus 18. In plain English, ABCA1 is a lipid export pump. It helps cells move fats and cholesterol out. That matters because dry AMD is connected to waste handling and lipid biology in the eye. So Robin did not only suggest a drug candidate, it helped point toward a possible biological mechanism. That is a huge difference. A search engine gives you links. A scientific AI agent can help build the next question. Then came the second round. Robin proposed more candidates, and researchers tested 10 of them. One major candidate was Ripasudil. Ripasudil is already clinically used as an eye drug for glaucoma in Japan. In the Robin paper, Ripasudil enhanced RPE phagocytosis in vitro and outperformed Y-27632 in the tested setup. The paper reports Finch's analysis as a 7.5-fold increase versus control, while a human analysis showed a smaller 1.75-fold increase. That difference is actually useful. It shows why humans still belong in the loop. AI can move fast, and human review keeps the speed honest. Nature's 2026 article also says Robin confirmed in vitro efficacy for Ripasudil and KL001. KL001 is another candidate from the tested list. This is not a proven treatment for dry AMD. These were cell experiments, not full clinical trials. But it is a very strong example of AI helping scientists become more systematic. The honest version is, Robin helped discover promising in vitro drug candidates and a possible mechanism for a hard eye disease. That is already impressive. We do not need to exaggerate it to make it exciting. The bigger story is the workflow. Robin closed part of the loop. Read literature, make hypothesis, suggest experiment, receive lab data, analyze results, suggest the next experiment. That loop is why AI in science matters. It can turn a giant pile of papers into a ranked research plan. It can help small teams move like bigger teams. It can help scientists spend more time testing and less time drowning in documents. Where this is headed is semi-autonomous science. Human scientists still choose the goals, run the physical experiments, check the results, and decide what is biologically meaningful. But AI systems like Robin can become the engine that keeps generating better next steps. In a future, a scientist might say, here is the disease, here is my lab setup, here is my budget. Find the most testable ideas. And the AI could return a ranked plan, a reading list, suggested assays, and an explanation of what evidence supports each idea. That is not replacing scientists. That is giving scientists leverage. Robin did not solve blindness, but it did show what a serious AI lab partner can look like. And for science, that may be one of the most useful AI upgrades we have seen yet.